Hey everyone, we're looking at microorganisms and the immune system in this lecture. And there are a variety of different microorganisms. And when I say microorganisms, I just mean anything that's a living thing that's small. Virins are a bit unique. So virins is plural for virus. And a virus on its own right can't reproduce. And reproduction, being able to reproduce is one of the essential qualities of determining whether we should call something living or not. And virins can't do that, and so there's a little bit of a question here about if they're considered living or not. But once they infect a cell, then they in fact can reproduce, and they are in that sense alive. So a little bit of a gray area here, but there's a whole bunch of different kinds of virins out there. There are ones that have a DNA genome in them, or they can have an RNA genome in them. Regardless of what the type of virus is, it will have some genetic information in it that codes for, gives the infected cell the instructions for how to build build more virus. And then it can also be enveloped or non-enveloped. And it can also be enveloped or non-enveloped. And this refers to, does it have a plasma membrane surrounding the portions of the virus that are the genome, or does it not? So those are some of the main characterizations there of all the different virins. A microorganism could be a bacterium, and there are a couple of different types of bacteria based upon their shape. So if a bacterium is spherical, like a ball, it would be called coccus, is the word. And coccus bacteria, here's a scanning electron micrograph that shows little individual balls. And I should say that bacteria are generally very small, like a very small fraction of the normal size of a normal cell. And the arrangement of the bacteria can vary too. So in like this example, we're seeing bacteria arranged in such a fashion that they almost look like little clusters of grapes in a way. That's called staphylo. So this particular bacteria then would be Staphylococcus aureus, I think is what we're seeing here. But Staphylococcus refers to that it's spherical shaped bacteria, and then they are clustered together in that grape-like cluster formation. So that's what makes it staphylo. Okay, you could also have a streptococcus, and strepto would mean that it's little spherical balls, but instead it's being in clusters, it's more like a chain. So it would look something more like this. This is Streptococcus pyogenes, the bacterium that causes strep throat. A bacteria could also be curved, and these are sometimes called vibrios. And it would be sort of the same convention as well. You could have probably a streptovibrio type of bacteria, but you can see they're like little rods that are slightly bent. You can have just rod-shaped bacteria that are more of a straight rod like this, and these are called bacillus. And you can also have like a streptobacillus type of bacteria as well. Okay, there are a couple of unique different kinds of bacteria. Like this one is called a spirochete, and a spirochete is shaped like a corkscrew, and so it gives it some interesting characteristics and mobility because it can literally drill in through mucous membranes and things into deep recesses of your body. This particular one that we're seeing here is Borrelia burgdorferi, which is the bacteria that causes Lyme disease. So a pretty nasty pathogen here. But by and large, most bacteria either be spherical or a slight curve to them, or just a rod. Okay, above and beyond then bacteria, there can be a whole host of other types of living things that could be a microorganism and might infect your body at some point in time. Here's a couple examples. This is Plasmodium. This is a microorganism that invades red blood cells, and it's the causative agent for malaria. This nasty looking thing here is called a schistosoma, and it's like this little worm-like creature that can get into different parts of your body and make you sick. And there's a whole bunch of other types of organisms like this that are neither a bacteria or a virus, but yet can still infect your body and make you sick. Okay, now oftentimes it's the case that people, when they think of bacteria, they think of bad stuff, stuff that makes you sick, stuff that harms you. But I do want to stop to point out the fact that a lot of bacteria are actually super good for you, and furthermore, pretty much make our world run. Bacteria are one of the most abundant living things on the planet. And for example, you have so much bacteria in your body that for every one of your body cells, you have 10 bacterial cells. So you're actually 10 times more bacteria than you on a cell-to-cell count. Now, that statistic's a little misleading because bacteria are really small. So even if they outnumber your cells 10 to 1, they're so small that their volume doesn't take up that much. But in either case, there's a lot of bacteria in you. Most of the bacteria that are in you live in your large intestine. This is called the microbiome. And the microbiome is going to do all sorts of things for you. It's going to create vitamins that you yourself don't actually create. So some of the big ones are vitamin B12 and also vitamin K. The bacteria within your gut will help to break down more complicated lipids, fat-type molecules, and help you absorb those. And the benefits of the microbiome can be so substantial that if somebody was given an antibiotic and it killed all of the microbiome and more of a harmful bacteria grew in the place of the normal bacteria that were there, it can actually cause somebody to be really sick and even like pass out and have low blood pressure. Sorry, excuse me. You can probably tell I'm feeling a little sick. Speaking of bacteria, but there's a condition, for example, known as C. diff, which is not an uncommon thing that happens in elderly people, and it's basically the proliferation of a more harmful type of bacteria that outcompetes the microbiome, and it makes people very sick. So bacteria are very beneficial to keeping you healthy, and furthermore, they're also being used and engineered to do all sorts of cool things. Like they're trying to make bacteria that can produce oil, so oil then becomes somewhat of a renewable resource that we can grow. Bacteria are used to produce different medicines, like human insulin is often produced by bacteria through some genetic engineering. They're working on trying to create bacteria that can break down plastics and make them easier to recycle. So they're actually a pretty cool group of living organisms, and I hope at least you'll come out of this lecture more inclined to not just revere them as pests that cause people to be sick and cause harm. They can do that, but there's a lot of really cool beneficial parts to bacteria. Alright, now let's talk then about how your body protects itself from a whole array of potential invasions of microorganisms. Your immune system is divided into two different parts. There's what's known as the adaptive part, and there's what's known as the innate part. And there are some key differences here. The adaptive part, you're not born with it. You actually develop this over time. And the innate part, you're actually born with this. And it doesn't develop over time. The adaptive immune system is highly specific, meaning not only could it pinpoint that your body's being infected by a pathogen, it could pinpoint that it's specifically a bacteria and specifically the strain of bacteria that it is. Very, very specific. The innate immune system is not specific. And so it could help you defend against some bad stuff, but it can't necessarily distinguish what the bad stuff is. It can't figure out if it's a bacteria or a virus or a parasite or something. Now, a couple examples of what your innate immune system is this is a couple of different things that are involved here. It could be barrier defenses. So these would be things like skin and snot and mucus and saliva and tears. Anywhere where there's literally a physical or a chemical barrier that keeps pathogens from just getting into your body, that would all be considered and categorized as part of the innate system. There are other things like inflammation would be considered a component of this. It's sort of an innate response that encourages white blood cells to get to a site of infection, so it helps to reduce the severity of an infection by changing some of the blood flow to different areas. There is this thing called the complement system, which is like this inherent intrinsic set of proteins that are dissolved in your blood plasma, and if they detect an infection, they can be rallied to precipitate out of solution, and well, maybe not precipitate out of solution, but they undergo a big enzymatic breakdown and a chain reaction of events that ultimately leads to destroying whatever the pathogen is. So it's a really complicated pathway. We won't go into it, but it does nevertheless exist inherent in your blood, this cool ability to activate this defense system and destroy anything that's in your blood that's not supposed to be there. So those are a few examples of the innate immune system. The adaptive immune system then is the part of your immune system that's highly specific, and it is going to have a couple different parts to it. There's what's known as the cell-mediated portion, and there's what's known as the humoral mediated portion of the adaptive immune system. And basically what this means is that one part of the adaptive immune system uses cells to kill the bad stuff. The other part of the immune system uses fluid, which is what this word means, humoral. And specifically, this is referring to antibodies. So in your adaptive immune system, you have cells that are each themselves can go and destroy the bad stuff, or they can create these things called antibodies, which then go and destroy the bad stuff. Antibodies, we'll see here in a minute, are a very strong immune response. As soon as your body starts making antibodies against an invader, it's only going to be a matter of time and before that invader is targeted for destruction and eradicated, hopefully, from the body. But on the flip side, if there's like a false alarm and you start producing antibodies that bind actually to parts of your own body, that can be really severe, and that would lead to something what we call an autoimmune disease potentially. And so it's generally this is a very strong response, and so it's highly regulated. Only a bunch of criteria, only if a bunch of criteria are met, will the body then employ the production of antibodies. So we'll talk a little bit about some of those. But regardless, the adaptive immune system, it relies on the help of lymphocytes. Lymphocytes are a kind of white blood cell that are like the warriors of the immune system. And there's two different types. Here's pictures of what a lymphocyte looks like in a micrograph of blood. Here are the two different main camps here of fighters. We have B cells and we have T cells. So this would be a T lymphocyte. This is a B lymphocyte. T lymphocytes, they mature in the thymus. So I sometimes recall that T stands for thymus. And B cells mature in the bone marrow. And B, coincidentally, helps us remember that's from the bone marrow, although I think that they're named actually from something else. It's not because they're made in the bone marrow. But in either case, these cells mature in different places and they have different jobs. So the T cells can either be what's known as a helper cell, or they could be what's known as a cytotoxic cell. And the jobs of these two are the following. A cytotoxic cell would destroy a cell that's infected with a virus, most often times. Okay, any cell that's infected with a pathogen, most likely a virus, would be detected and destroyed by a cytotoxic T cell. A helper cell, a helper T cell, its main job is to help activate a B cell. And if it does activate a B cell, what a B cell does is it goes on to convert into this series of cells that create antibodies. Okay, so the B cells have the potential to be very powerful cells. They can create antibodies if the proper triggers are pulled. And again, that's a really strong response. Now, how do these T cells and these B cells work? It has to do with recognizing what's known as antigen. And it has to do with this thing known as antigen presentation, which we'll talk about in just a minute. Before we do that, I'll just provide a quick survey here of some of the important organs that form the immune system. We said that the thymus and the bone marrow and the cells that they make here are very important, but there's also some other structures too to consider when we're talking about the immune system. So some of the ones that would be involved, bone marrow, thymus, but then above and beyond that, there's a whole network of lymphatic vessels, and lymphatic vessels are continually taking samples of fluid from around cells, and they are screening it for pathogens. And so they'll take that fluid and they'll pass them through a whole network of what are called lymph nodes. And basically how it works is if this is a lymphatic vessel, the fluid that it's collected from your body will pass through these tubes. It will then enter a lymph node, and there's a whole bunch of B and T cells here in the lymph node. And so if there's any sort of invader in any part of your body somewhere, it's likely going to get taken up in this little collection of fluid, and at least a part of those invading, or a small portion of those invading particles will likely get taken up. And then they'll have to be passed through a whole garrison of these B and T cells. And those B and T cells can recognize pieces of molecules and pieces of cells. And specifically, B and T cells are trained to recognize only bits and pieces that are not supposed to be in the body. So they are very familiar with all the different fragments and bits and pieces of cells that are supposed to be in your body. We call those self-antigens. But they are trained to seek out and destroy any that are non-self antigens, pieces of fragments of bacteria or virins or other microorganisms that are not supposed to be in your body. Those are the things that they detect and destroy. So there's a greater chance of recruiting B and T cells to fight the good fight if there's this fluid that's passing through all of these thousands upon thousands of B and T cells. If anything gets picked up in your body, at some point it will likely pass through a lymph node, and it will pass through all of these cells. Now I should say that each cell, a B and a T cell, are trained to recognize a very, very specific chunk of stuff. We call it an antigen, a very specific antigen. And so it could be like, let's say you were infected with a virus of some kind. It could be that that virus got into your lymph and pieces of it were shown to all of these cells, but maybe 99.999% of them didn't recognize the piece of the virus because these cells are trained to recognize only a very specific thing. But fortunately for you, there's so many of these cells that recognize so many different particles that the chances are, if some virus gets into your body, that at some point it will bump into the 0.0001% of the cells that do have a receptor that matches and can bind and detect the foreign thing. And if that's the case, it's going to start an immune response. Okay, tonsils and adenoids are similar to lymph nodes in that there are places where there's a lot of B and T cells, but they're not necessarily connected to the lymphatic vessels. They are situated, though, in a strategic place right by the throat, whereas a lot of stuff is coming in and out. So they're monitoring everything coming in from the nasal cavity and from the oral cavity. make sure there's nothing that's going to harm you there. Okay, the appendix is another place. There's a lot of B and T cells waiting here in a strategic spot because it's somewhat of a gateway between the large intestine and the small intestine. And the large intestine is the place where most of the microbiome exists, and the small intestine is generally relatively sterile. So it is a strategic place for there to be B and T cells on hand at that gateway between the part of your body that has a lot of bacteria and the part of your body that doesn't have very much bacteria. There are Peyer's patches. These are similar to tonsils, except they're found in the along the intestinal tract. And then lastly, the spleen is an organ that, similar to how lymph passes through lymph nodes, and the lymph nodes will screen the lymph for pathogens, the spleen also screens the blood for pathogens. All right, let's talk then a little bit about what's known as antigen presentation. This is what is the crux to the whole process. This is what allows your immune cells to recognize that you are being invaded by a pathogen, and then to turn on some immune responses to counteract that. Okay, antigen presentation relies on the concept that, or the nature of a cell that cells will put pieces of themselves out on the surface of the cell. Nearly every cell does this. This was a cell here. They have these little flagpoles, and they put little pieces of themselves on the flagpoles. And this is a normal thing that happens. Cells are good at doing this. And the reason they do it is to help communicate with other cells. They help to show other cells in the body what kind of cell that they are. And it's a very natural, normal, and important thing. Now, the flagpole that they actually present pieces of themselves on is called MHC class 1. And there's another type of flagpole called MHC class 2, which is also very important, but we'll get to that in a minute. Okay, so let's say a virus was to wiggle its way into this cell. So here's my little virus here, and just in the same fashion a cell is used to portraying anything that's inside of it on the outside, little bits and pieces of anything that's inside of it, put it on the outside so the cells can have a window in, see what's going on inside of the cell. Well, that also is the same fate for a virus that comes in. Eventually, the cell will break down some bits and pieces of it, and it will put those bits and pieces, same as it does with everything else in the cell, it'll put those bits and pieces on a flagpole, which is called MHC class 1. And now we've got some little virus particles out on the cell as well. And there could be a whole batch of different particles, you know. Well, each of these characteristic little parts of the virus that can be recognized are called antigens. In fact, any antigen is a general word that means anything that is a recognizable pattern on a molecule that something could bind to specifically. So all of these things are antigens on the outside of the surface of the cell. And specifically, though, these antigens would be what we call non-self antigens. The rest of these are called self-antigens because they're bits and pieces of things that are supposed to be in the cell. Whereas this little viral particle, this is called the non-self antigen, and this is the one that's going to gain the attraction and the attention of the immune cells. All right, so what can happen then? We can have one event occur where we have a T cell, and this would be a T helper, or sorry, a cytotoxic T cell. Cytotoxic T cells, cytotoxic T cells are trained to grab on to any non-self antigen that is presented on MHC class 1. So this means any cell that's been infected by a pathogen or invaded by a pathogen. And if that's the case, if it binds this non-self antigen, it will probably it will not bind any of the self antigens, and the reason for this is it undergoes a lot of training in the thymus to not do this. In theory, only the cells that leave the thymus, only the T cells that leave are those that would bind only non-self antigen, not any self antigens. It'd be bad if you had T cells going around and binding any cell that had self antigen, because all cells have self antigen on them, and then that cytotoxic T cell would be breaking down all of your cells. We don't want that. So cytotoxic T cell, as the name suggests, cytotoxic means toxic to the cell. Cell means, or cyto means cell, and toxic means toxic. So this is a cell that is going to be toxic to another cell, but specifically one that has non-self antigen. In other words, any cell that's infected with a virus and is displaying pieces of that virus on the surface, those can be recognized by a cytotoxic T cell, and that cytotoxic T cell will destroy the infected cell. This is called cell-mediated immunity, because the killing of the infected cell is done by another cell, namely a cytotoxic T cell. Okay, there's another scenario though that can play out, and this happens when we have what are known as professional antigen presenting cells, or PAPCs. These are a special kind of cell that also present antigen, same as all of your other cells, but these guys here can professionally take in a pathogen and professionally present its antigens. And we won't get into the complexity here of how all of those mechanisms work, but the biggest thing to know is that a professional antigen presenting cell would be a cell that can engulf a virus, engulf a bacterium, engulf a small parasite, any pathogen, and that is a different situation than what I just described earlier. When it engulfs the pathogen, it's not being infected by the pathogen, it is actually professionally handling that pathogen by professionally absorbing it. Well, when this is the case, and it's not actually infected by the virus or the bacterium, but it grabs onto it and eats it intentionally, it knows how to present those bits and pieces in a special way. Okay, so if this was a professional antigen presenting cell I'm showing you here, there would be on it self antigens, just the same as all other cells. And those are all on this flagpole that's called MHC class 1, just as we saw in the previous example. And so there's all these little antigens, different bits and pieces of the cell presented on the surface of the cell to show other cells what kind of cell it is. But here's what makes a professional antigen cell a professional antigen presenting cell, not just a normal cell. It will take anything that's absorbed. So let's say we've got a little bacterium in here. It absorbed that bacterium. That bacterium didn't infect it. It actually sought the bacterium out, targeted it, and consumed it intentionally. Then, if that's what happens, the pieces of this bacterium will still get put out on the surface of the cell to show the whole body what it's found. But the thing that's the main distinction about how this is presented is that this is not presented on an MHC class 1 molecule. This is presented on something called MHC class 2. And the note here would be, you could think of it this way, that anything presented on MHC class 2 suggests a pathogen that has been professionally, quote unquote, we won't really get into what it is that deems that, but absorbed. Okay, so that's what makes a professional antigen presenting cell unique, is that it can express antigens on this unique flagpole, which is called MHC class 2. Now, MHC class 2, this antigen presenting cell will go through, probably get somehow into the lymph, go from lymph node to lymph node to lymph node to lymph node, and show all the T cells it can and show all the B cells it can these little fragments that it found. And what it's doing is it's looking to find the right immune cells that have that recognize these bits and pieces. We said that T cells and B cells, they all have these little receptors that bind to antigen, but they are very specific in what they bind. So it could be 99.9% of all the cells that are seeing this antigen don't recognize it, but there will probably be some random cells in the body somewhere that have the right receptor that recognize this, and it's all totally random. So if we're using a professional antigen presenting cell, what generally needs to happen is we don't want this cell to be destroyed per se, because this is an immune cell. It's a professional antigen presenting cell. Instead, we want to use these antigens to turn on the antibody mediated immune response. So what needs to happen is there has to be a T helper cell that comes and recognizes these antigens, and then that T helper cell will become activated, and then it goes and turns on a B cell. And when the B cell is activated, the B cell creates antibodies. In a very simple way of thinking of it. This all is actually highly complex, but we won't get into any of that level of complexity. Okay, so for the exam, this is sort of the distillation of what you have to keep in mind. If we have antigen, and it's specifically non-self, so it's antigen from things that are not supposed to be in your body, if it's presented on MHC class 1, sorry for the chicken scratch, MHC class 1, here's the meaning of that. It generally means a cell that is infected with a virus. And so then there's going to be an outcome of this. Generally the outcome is the infected cell is destroyed by a cytotoxic T cell. Okay, that's one side of the coin. This is all called cell-mediated immunity. It's all part of the adaptive immune system. Okay, another scenario then is that you could have non-self antigen presented on MHC class 2. Okay, that's the big difference. MHC class 1 in the first scenario, MHC class 2 in the second scenario. What that means, what that signal to the body is roughly this. A cell has professionally consumed a pathogen. And then the outcome is different. We don't want the cell that's presenting the antigen then to be destroyed. We want that a different thing to happen. So what happens is the professional antigen presenting cell turns on a T helper cell, and the T helper cell turns on a B cell. The B cell then creates antibodies in a very simplistic scheme here. This is sort of the boundary. This is all part of humoral or antibody mediated immunity. And I think we'll leave it at that. Okay, two different sides of the adaptive immune system. You got non-self antigen presented on MHC class 1. What that means to the body is that that cell is likely infected, and so it's destroyed by a cytotoxic T cell. You have antigen, non-self antigen presented on MHC class 2. Well, that means is that it's on a professional antigen presenting cell that has professionally digested and absorbed it, so don't kill that cell. Rather, let's turn on a more robust immune response. So a T helper cell would likely get activated by that antigen presenting cell, and then later a B cell would become activated, and then it would start creating antibodies. Antibodies are a very strong immune response. All right, let's talk then a little bit about why antibodies are useful. What do they get used to do? Why are they effective in curtailing an infection or an invasion? Here's an antibody. Antibodies have several parts to them, but there are 2 binding sites on an antibody. And the other thing to note about these is that these are highly selective. So these binding sites are going to bind to like one thing and one thing alone, likely. Maybe a handful of things, but generally it's just one thing that they will bind to, which is good, because if we create antibodies to destroy a pathogen, we want them to bind to and destroy the pathogen and the pathogen alone, not any other similar type of molecule that's supposed to be in your body or something. Okay, so that's one thing to note. And then the other thing to note is that there are 2 binding sites here, which can help create some interesting attributes here. So here are some of the things that antibodies can do. The first is agglutination, and this is what utilizes those 2 binding sites. If you have a bunch of molecules with 2 binding sites, what they can do, let's imagine I've got a bunch of these are little, let's say these are a bunch of spherical coccus bacteria here. An antibody could bind to this, and then it could also bind to this and form a structure that looked like this, where you have the antibody and the 2 pathogens. Well, then you could have another antibody come into the scene, and it could grab onto that clump and then grab onto another free-floating thing, and then you would have you start to form an immune complex that has pathogens glued together, and the glue is the antibody. And over time, if this continues to occur, within a matter of seconds, honestly, all of these pathogens, if they're in a nearby place and there's antibodies on hand, they would turn into a big clump. With the antibodies being somewhat the glue that holds it together. That's called agglutination, and why it's a powerful response is that it's easier for the body to get rid of one big clump of bad stuff rather than to go chase down and try and destroy a whole bunch of individual tiny little microscopic things. So it's better you clump it all up. It's easier to get rid of that and target it for destruction and get it out of your body. So that's a powerful response there. Another thing that antibodies can do is they can neutralize things, and basically what this means is that the antibodies block receptor sites or toxins. So, for example, a virus might have some receptors on it that allow it, here's some epithelial cells, some cells lining like maybe the inside of your lungs or your skin or the inside of your esophagus, your mouth. Well, it's usually the case that a virus has to bind first to a cell, and it's usually that there's some sort of receptor on the cell that actually is going to bind to the virus, because the virus sort of mimics. It has a receptor on it that's similar to a human receptor, and so it allows it to the body gets tricked into inviting it in. Okay, it can actually attach to the receptor, and then it gets in and wiggles its way in, and now if it's in the cell, it can start hijacking all of its cellular machinery and use it to replicate itself. Well, if we could get some antibodies in here to plug up all of these receptors on, let's say, the virus, then the receptors can't bind to the doorways of your cell. So that's called neutralization, when the antibodies cover the virus and block it from binding to anything. Similarly, if a bacterium created some sort of toxin, and the way that the toxin worked is that it bound to different things in your body and caused destruction. Well, if you had antibodies against that toxin, the antibodies could actually bind to the toxin, and then the toxin can't bind to the things that it normally binds to make you sick. So it can just prevent toxins and viruses and things from doing what they normally do. Okay, opsonization, this literally means to prepare to eat. And what this means is generally a cell, when it consumes, let's say here's a little bacterium here. When a cell consumes a bacteria, that process is known as phagocytosis. It literally eats it. Well, some of these bacteria are slippery little buggers, and they're hard to grab onto, and they're hard to eat. So antibodies can bind to them and almost become like little handles that allow the phagocytes to phagocytize them, to eat them. So that's what opsonization is. And then also antibodies can stimulate the inflammatory response. And this is good, again, because anywhere where there's inflammation, there's generally increased blood flow to that area, and that allows more white blood cells to get to the site of tissue damage and infection, and then they can get in and fight the good fight. Now there are a couple other functions to antibodies too, but we won't talk about those in this class. Okay, so then keeping in mind some of these things, this whole business about natural immunity and vaccination, this is a major contentious issue in the COVID-19 pandemic. You were hearing things about herd immunity. You were hearing a big battle between how what's more effective, the vaccine or actually getting the virus. A lot of controversy over that issue. Well, there's no mystery here. There's absolutely no doubt, no question about it, that natural immunity is always going to give you a much more robust immunity than vaccination. Anybody that doubts that just has a very little working knowledge of immunology. But the problem is that natural immunity can be actually pretty costly because you're actually getting infected. It's sick. So the controversy shouldn't be whether which was more effective, getting a vaccine or actually getting the virus. There's no question there. Naturally getting the whole virus infecting your body is going to give you much stronger immunity. No question. But the question that is the one that people should debate is, to what extent is the damage worth it? Because if you get infected with a virus, you're actually going to get sick. And depending on how severe the infection is and how severe the illness is, that could mean like permanent damage or even mortality. And so that wouldn't be worth the cost. Then in that case, the vaccine's a far better option. So that was just for your sake, that's an important thing to keep in mind. And that is the question worth debating, not whether or not natural immunity is stronger than vaccination. There's no controversy there. That's a settled dispute. And here's why. Here's an example of COVID-19, the virus that causes COVID-19. So this is an illustration of SARS-CoV-2. Okay, SARS-CoV-2 has a couple different proteins on it. It has an envelope protein on it. It has a membrane protein on it. It has a spike protein on it, and it has a lipid membrane on it, and it has some of these nucleocapsid proteins. Okay, so there's different parts and pieces to it. If this thing gets into your body, your immune cells are going to be binding to all different parts of this thing and causing immune responses to lots of different portions of it. Okay, these are all antigens because they're all recognizable portions of the virus that your immune cells could recognize and mount an immune response against. And the more different components you give the immune system to mount an immune response, the greater your immunity is going to be. Because instead of just one or two immune responses that are generating immune cells, you have a lot of different immune responses generating immune cells. So there's much more what are called memory cells on hand. Memory cells are cells that are on hand to detect the same invading pathogen and detect it much faster, so that in a subsequent infection, it gets destroyed. This is why we were seeing a lot of people that got vaccinated. They were having, pretty strong infections with SARS-CoV-2 later, and people that didn't get a vaccine, they were more or less more protected, okay, because they had more memory cells than the people that got the vaccine. And here's why that was the case. The vaccine, all it is pretty much this, as far as I understand it. There's different kinds of vaccines, but as far as I understand, when you get a vaccine, you're just getting this one little part of the virus, and it's safe because you're not getting the whole virus. It's not making you sick. You're just getting this one little piece of it. But at the same rate, by the same rate, you're only creating immune cells that recognize this one little thing. And so they'll recognize it, they'll stick around for a while, and then they'll leave, and then now you're more susceptible again, and so you'd have to get like a booster shot or whatnot or whatever. Now, then if you are infected with the virus, you're going to be making far more immune memory cells because you're creating an immune response for each of these little fragments and portions. So there's a much longer lasting immunity, much more robust immunity. So that's sort of the way to think about it. And I was a little perturbed about how much crisscross thing there was in the information and all of the, this is very simple immunology 101 stuff, and it was just sort of appalling to see how little of these very simple fundamental ideas were being conveyed during that whole chaotic time. Now, I'm not going to say either one way whether people should get vaccinated or not, because again, it all depends on the cost of how severe an infection could be, and is that severity worth the greater immunity. But there's absolutely no question natural immunity going to be stronger than infection with the actual virus is going to be stronger immunity than the vaccine, for sure. So for example, like if people did have a positive test and they did actually in fact have a COVID-19, there should be very little incentive for them to then go get vaccinated, because it's like they've already seen not only the spike protein, which literally is the vaccine, but they've also seen all of these other things. So they've gotten like a, if they were able to get through an actual infection of the virus, they got the vaccine. That's this thing here. But they also got all these additional parts. So it's like a quadruple powered vaccination. And I was very alarmed, and I hope that you would be too, at how strong the mandates or how strong the propaganda was to try and get people that have already been infected with the virus to then go get a vaccine. And that makes absolutely no sense, in my opinion. And when you start seeing that type of stuff, it makes you wonder what the heck is in that vaccine. Why is the push so strong for people that have already gotten the virus and they've already become immune to it? So that's my little soapbox tangent. I'll leave it at that. I won't go too far into that. We've already had a lot more than our fair share of controversy and crosstalk on this issue. So I'll just keep it at that. Okay, but the one thing I'll point out is that this is not a new issue, right? This same issue was something that presented itself in the mid-20th century about the polio vaccine. So there's two different vaccines. There was the Jonas Salk vaccine, and this was a portion of the virus. It was a killed virus, so it wasn't alive, and that was the first vaccine that was put onto the scene, and it was good for a while, and the United States was using it, and then Albert Sabin created a vaccine that was it's called a live attenuated virus. So that means it literally is the actual virus, but it's just been weakened in some way, so it's not so virulent. Well, this vaccine was not being used in the United States because the United States was already pretty much sold and committed to producing the Salk vaccine, but Sabin partnered with the Soviets, and they started cranking this thing out, and the rest of the world was using this vaccine while we were in the United States using this one. And it was showing that the Sabin vaccine was much greater, conferring a much stronger immunity. And so then people started to wonder, hey, why are we using this Salk vaccine when the rest of the world is just using this stronger vaccine? But the problem was that the Sabin vaccine was actually giving people polio because they were, even though the virus was attenuated, they were being given the actual polio virus. So there was like maybe one in every, several million cases where people were actually coming down with a form of polio. And I don't know if it was the severity of it was reduced or if it was still a pretty robust case. But in either case, that was sort of the controversy. And so in the United States, we did then end up switching to the Sabin vaccine, and then I think we switched back to the Salk vaccine. And so now there's still both versions of them are still both used, and the general consensus is that the Sabin vaccine is good in areas where polio is still rampant. It gives a very strong immunity to a population. And in places where the polio is mostly eradicated and it's mostly just, monitoring things and keeping things that way, then the Salk vaccine is preferred. That's my understanding. But there's an interesting video that we watched in class that talks a little bit about this. Or there's an interesting article we read in class that talks a little bit about this dilemma, and it's related to some of the stuff that we had experienced with the COVID-19 pandemic. Okay, there we have it. I hope you have a better appreciation and understanding for a little bit about what's going on underneath the hood of your immune system and some of the factors at play. Again, very simplistic overview, but I think good enough to give you a primer, at least in the immune system.